So good morning, good afternoon, and good evening to each and everyone present over here. I, Dr. Tirthraj Singh, on behalf of Center of Excellence in Information Technology, Health Informatics, Department of Biotechnology and Bioinformatics, Jaypee University, Solan, Himachal Pradesh, India, welcome you all on this 6th webinar of this IWS, International Webinar Series of this center. We are organizing this since August continuously, every month, one webinar. We started with the very first webinar delivered by our Honorable Vice Chancellor, sir. And this is the 6th in the series. And we are planning to keep it like this is the last one for this particular season. So we are calling it as end of the season 1 today's webinar. And we are having one of the eminent academician and researcher, Professor Holger from Fraunhofer Institute and Bonn University, Germany. So before moving further, first I would like to call upon our head of the department, Professor Sudhir Kumar, to first welcome Professor Holger and brief the gathering.
Good morning, Professor Holger.
Thank you very much. And yeah.
And my greetings to you for this new year. And I hope that everyone is safe. All the participants, you in Germany, our alumni in Limansa, and all the participants. Greetings from our side, from the Department of Biotechnology and Bioinformatics. And I formally welcome Professor Holger. And thank you so much for giving your time, effort, and agreeing to give this webinar that is actually conducted by one of the Center of Excellence in Informatics and Healthcare, Dr. Tirthraj, Dr. Ragini, and Dr. Nishant, a very active member of this center. And I'm very hopeful, Professor Holger, that your talk will be very useful to all of us. And I also see that this is like a beginning for a relationship and a collaboration between your university, your group, and here in India in our bioinformatics research group. So I also feel once this COVID pandemic is over, you will physically be here with us, visit us, and we will have deliberations for mutual research projects, funding opportunities, and students' projects. So many sincere thanks to you, and we look forward for your presentation now. Thank you so much.
Yeah, thank you very much for the nice introduction and for the honorable invitation also to give a talk to you.
Okay, Professor Holger, just a minute. Let us introduce once more with great detail, because you are having a very great profile. So I would like to call upon Dr. Ragini to introduce you formally, and then we will start your talk. Dr. Ragini, please.
Yeah.
So hello, Professor Holger.
Hello.
So hello, everyone. Today, as you already know, that Professor Holger is going to present a very, what we can say, the important and affluent talk on a very imperative and what we can say the currently important topic. So I will introduce about Professor Holger. So Professor Holger is presently Head of Artificial Intelligence and Data Science at Fraunhofer SCAI and Professor at University of Bonn, as Dr. Tirthraj has already mentioned about this. So what he's doing currently, he's responsible for a newly formed artificial intelligence and data science team that is focusing on precision medicine and early drug discovery. So basically, he's a computer scientist by training, and he's having a deep interest for life science and medicine. Currently, he's working within the intersection of data sciences and medical application fields. He strongly believes that data-driven methods together with domain-specific knowledge can bring a real benefit for science and for patients also. So he's specifically interested in developing and applying advanced data analytical, for example, data mining and machine learning, and artificial intelligence techniques for questions in modern biomedicine, specifically in precision medicine and early drug discovery. So now I will talk something about his education. So he did his master's in computer science from the Philipp University of Marburg in 2002. And he's having a specialization in artificial intelligence, data mining, machine learning. And he did his master thesis at Max Planck Institute for biological cybernetics. He did his PhD again in computer science from Eberhard Karls University, Tübingen from 2003 to 6. Specifically, he worked on the machine learning with kernel methods, chemoinformatics, and bioinformatics. Hereafter, he did his postdoctoral research from DKFG German Cancer Research Center, Heidelberg, Germany, from 2006 to 2009, where he worked on a statistical analysis of biological high-throughput data, machine learning methods for learning biological networks, and machine learning methods in precision medicine also. Later on, he was the senior scientist in Cellsome AG, Heidelberg, Germany, where he worked on chemoproteomics data, machine learning models in chemoinformatics. After 2018, he's a honorary professor in the University of Bonn. And during 2010 to 2015, he was the group leader and associate professor for algorithmic bioinformatics in University of Bonn. During 2015 to 18, he was the principal lead analytics scientist in Monheim. During 2016 to 19, he was the adjunct research group leader in biomedical data science. From 2018 to 19, he was the director, head of data science and Lament. And very important thing is that he is having very good publications in internationally reputed journal with good impact factors. And he is having more than 120 publication titles, and that all publications are peer reviewed. So now I would like to invite Professor Holger to deliver his talk. Today, he is going to share his expert opinion on the very essential topic, that is multimodal data integration in biomedicine. So I'm sure that this talk will be advantageous and enlightening for all of the participants. So now I would request Professor Holger so he can start his imperative and beneficial talk for all of us.
Yeah, thank you very much again for the very nice invitation and summary also of my CV. And again, so I feel really honored by this invitation, and I really hope that indeed after the pandemic situation has become better, that it would be possible to meet physically also, and then also discuss maybe further collaboration opportunities. And by the way, sorry, so that I think there's sometimes a time delay in the voice, so that I unintendedly at some point potentially interrupted you. So that might also in the future occur, so because of these time delay issues. So what I wanted to talk today about is a little bit about my work in multimodal data integration, specifically in biomedicine. And before I move into this topic, I just wanted to give you a very, very brief overview about actually the organization that I'm mainly working for. So I have two affiliations. One is Fraunhofer and one is the University of Bonn. So what's Fraunhofer? So it's in essence one of the world's largest non-profit research organizations. And indeed, it is also up to a certain degree spread all over the planet. So there we have also offices in India, in fact. And one of the focus areas, in fact, of Fraunhofer, and that's why I'm also there, is health and environment. The campus where our institute SCAI is located is in Birlinghofen. So this is close to Bonn. So it's in fact a suburb of Bonn. And it's in fact one of Germany's largest research centers for applied computer science and mathematics, with 3 research institutes sitting there. So one is indeed here SCAI, where we belong to, and then there are 2 others. And in fact, we have also our own castle here on the campus. Yeah, so what my team is doing at Fraunhofer is can be maybe summarized under the mission: bringing better treatments to the right patients. As you already also summarized a little bit. One of my ambitions really about data science and AI is to bring a certain benefit to patients. And therefore there's an application focus, a dedicated application focus on one hand on precision medicine and on the other hand on systems medicine and drug discovery. And of course, when you want to develop computational approaches in this area, so this oftentimes then requires also to develop dedicated methods. And this is basically the second pillar of what we are doing. So method development for solving particular application problems. And these specifically, indeed, as also mentioned before, on one hand, comprises combination of human knowledge with data-driven AI, which is sometimes nowadays called hybrid AI. We deal a lot with time series modeling, and I will give you a short glimpse also on this type of work. And indeed, also what I find quite important, and what is also indeed in the title of this talk, is this multi-modal and multi-scale data fusion. Because indeed, if you think about biomedicine and the complexity of diseases, this is rarely reflected only by one data modality. And at the end of the day, this is a kind of ping pong game in which we indeed then bring these methods back to solve dedicated application problems. On the other hand, these applications oftentimes then call us for developing particular methods. And we at Fraunhofer are specifically positioned to interface also with industry in the application field. So we are collaborating with pharma, with biotech, also partially with hospitals, and on the other hand, of course, also with other academic partners. And so this is really a bridge role, so which is really the strategic aim of Fraunhofer. Altogether, what we do can be also summarized via a certain value chain that we cover, which goes from biological system reconstruction and modeling over drug target identification, precision medicine, up to clinical trial design and simulation of synthetic cohorts. So to motivate just a little bit why I'm actually doing what I'm doing, because I'm not sure that everybody here in this talk really knows about all of these different aspects. So I guess there are also a number of students in this talk here, is the situation in healthcare. And in fact, what you can see here on the left-hand side is the fraction of money spent on the gross national product in Germany for health. And you can see that this is during the years 97 till 2017. Over these 30 years, and 20 years, in fact, almost steadily increasing over time. And on the other hand, you have to see another part of the picture, and this is the right-hand side here. So this is a figure taken from an article in Nature published in 2015, in which the authors, in fact, investigated the efficacy of different drugs in real world. And you see what is here exemplified here by these colors is the fraction of people who actually benefit from a given treatment. Obviously here this is stratified here by different indication areas, which are non-trivial. So we are not talking about a cold here or so, right? So, but about serious diseases. But what is really overall here, the worrisome picture that you get here is that, in fact, a larger fraction of patients indeed does not really benefit from these costly treatments that we prescribe to them. So we have, on one hand, increasing healthcare costs, and on the other hand, a quite substantial fraction of people for which these expensive treatments are actually not really effective. So likewise in the pharma space, you can also see a certain picture. And this is indeed a large fraction of clinical studies that fail. So in fact, larger than 90%, in fact. And on the other hand, a declining efficiency, also R&D efficiency in terms of money of biopharma companies. And this is, of course, quite challenging for these companies here, as you can imagine. So without going into too many of the details of the situation in pharma, one of the questions, of course, then that comes up to mind is what can we do about this situation here overall? So obviously one of the reasons behind, and there's of course also other reasons, but one reason behind this picture that I showed you before in these two slides is that indeed patients respond rather differently to treatment. So, and this has to be really seen in the context of the fact that the way that we currently think about diseases is a concept that dates back to the 19th century in essence. So it's purely phenotypically defined and it's likely outdated. So in that situation, then indeed the idea comes into mind rather to treat every patient that is diagnosed with a particular disease in the same way. Let's try to at least stratify patients into more homogeneous subgroups. And this is indeed what is meant by precision medicine. So the idea is that you, of course, do this at the end of the day with some sort of algorithm. So which then in essence would demix this heterogeneous population in some way. And then you would be able to find better tailored subgroup specific treatments and therefore, of course, also have a higher chance to see a benefit for patients in these dedicated subgroups. And that, of course, by the way, without going there into too many details, would have also an impact on clinical trial design in pharma industry and so on. So now, of course, the question arises, how can this be achieved in practice, patient stratification? So there are certainly different ways how to do this, but one of the possible ways is to think about a molecular based stratification of patients rather than on a phenotype based one, as I said, which is largely outdated nowadays. So now when we think about molecular data, then at the same time we have to see the fact that the etiology of many diseases is not covered by one instance of biological data. So let's say transcriptome data alone or genetic variants, but really with a whole spectrum of these data. So one has really to see this complex interplay over different biological scales. And one example, of course, in this context is cancer, which at the end of the day affects, of course, or starts from certain somatic mutations, but affects, of course, also epigenome, transcriptome, proteome, and eventually even the metabolome. And if you think about now patient stratification, then the idea is that comes into play more and more now into literature is not to base such a stratification on one of these data modalities alone, but really on multiple of them together, so multi-omics data. So hence covering these different biological scales. Of course, this is not without challenges because from a data science perspective, these different types of biological data have rather different statistical properties. So for example, think about genetic variants, which are often described as ordinal data versus something like transcriptome data, which lives on a continuous numerical scale. And on the other hand, of course, also the statistical distribution properties differ widely between these different data entities. And furthermore, what one should not forget is that there's also a highly different number of features simply which can really be different in orders of magnitude between, let's say, proteomics data, which is oftentimes only focusing on selected proteins, and on the other hand, let's say, genetic variants, which in case of SNPs might cover millions. So what we try to do now is to develop a approach which allows to stratify patients, in fact, based on multi-omics data and address some of these issues. And this approach, which we developed, we call pathway-based multimodal sparse autoencoder, in short PathMe. And before I go into more details, let me just give you a very brief overview about the concept behind. So the concept behind PathMe is really to use many different data modalities. And here on the right-hand side picture, these are actually only examples, and map the features, in essence, the different measured features of each of these omics sources to, let's say, pathways. So basically, certain knowledge graphs, let's say, in general, which reflect some of the insights from, for example, literature that we know is relevant in the disease context. So, and then with the approach that we have, so then we are able to estimate a per patient score, in essence, for each pathway, a summary score based on all of these different data modalities and features. So what can we do now with this score? So, well, one thing is indeed we can do patient stratification. So in particular here, we can use a so-called bi-clustering of patients. So that means each patient at the end of the day can be represented by a profile of different pathway scores. And then we can use certain techniques, so-called consensus sparse non-negative matrix factorization, for example, to identify subgroups of patients, as well as most describing pathways, in essence, so overall features, in essence, for each of these clusters. And this approach, in fact, PathMe, of course, we also compared against competing methods like SNF and iCluster, against which demonstrated, in fact, in a statistical evaluation, a favorable performance. So now let me explain a little bit more detail about what's behind of this method. So in essence, the background of this approach here is the framework of an autoencoder network. So an autoencoder network is a special type of neural network, which in essence here on the left-hand side here takes as input certain features, n different features. So let's say could be let's say 1,000 genes even, or whatever. And then on the right-hand side, it also produces features. And in the middle, however, and this is the real interesting part, so there's a compression of the data into a latent space. So that means there is an encoding step of the data, and then there is a decoding step of the data. And this entire transformation of the inputs to the reconstructed, in fact, output is something that can be non-linear. So each of these nodes here in this graph here are actually computational units, which have a kind of a nonlinear activation function here. So oftentimes one uses such a sigmoidal curve, and the edges here have certain weights, which are at the end of the day then trained. So at the end of the day, this is a model which is trained to basically compress data to a lower dimensional space. And this can be done, in fact, in such a way that it's also noise tolerant. So, for example, one can artificially increase input noise to increase the robustness of this procedure. So in our case here, we extended this original framework in order to come up with an architecture for multi-omics data. So, and more specifically, how this looks like here is depicted on the right-hand side. So in our case here, this encoding part, so the part where the original data is compressed together, in fact, here, consists in this case here of different modalities. So there's a certain number of features from gene expression which map to a certain pathway of interest, of let's say DNA methylation, so CPGs, of CNVs, etc. And at the end of the day, so then these are weighted against each other and compressed into this pathway score. And in a decoder, of course, they are decoded back into their original space. And all of this is, as I said, so this is a machine learning model. So this is trained. So it's data driven at the end of the day. And of course, one has to there also be a bit careful with respect to overfitting. So we want to have a representation, a pathway score that is in a certain way generalizable, and for that purpose we are using a number here of tricks that can be done. So for example, we use so-called input dropout. So that means the entire representation can be made sparse. Only a part, in fact, of the input features contribute at the end of the day to the overall pathway scores, and irrelevant parts are filtered out. And there's also a way how this can be achieved via a group-specific weight decay. And furthermore, we are using also certain approaches to account for the numerical range differences that exist, for example, between DNA methylation data and SNVs, which I mentioned. So there's a technique which is called batch normalization, which we use for this purpose. So at the end of the day, what this approach here does is, as I said, so it takes as inputs really here gene expression data mappable to a certain pathway of interest, and then learns a pathway of, a patient level multi-omics pathway score. So this is here the middle part at the end of the day. So now what you can do is since we have not only typically one pathways, but several pathways, is we get such a profile, a pathways score profile now of every patient here. So this is here exemplified here by this figure here on the right-hand side here. So this is results can be thought of a data matrix here really then all together, in which you have a number of patients, and each patient here has such a pathway score profile. And now what one can do with such a data matrix is one can apply non-negative matrix factorization to it. So what does this mean? So it means in essence that this data matrix is algebraically decomposed into two other matrices. So one which in essence reflects a contribution of different pathways to each cluster. So in this case, for example, there are 4 different clusters. So these are here the columns in this heat map, and here the color code here indicates the level of contribution of each row here, in this case a pathway, to this particular cluster. And on the other hand, you get this other matrix here. So this is the right-hand side, the so-called coefficient matrix, which indeed then shows in the color code here a soft clustering in essence of patients. So basically the higher, the more red the color code, the higher the likelihood that a particular patient belongs to this particular cluster. So this entire thing can also be made sparse, so that in essence as few as possible basis components are used to describe each particular cluster, and then this results into so-called biclustering. And furthermore, what one can do is to not only do this once, but in essence, with different random initialization, do this 100 times, in fact, or 500 times in our case, and then do a so-called consensus clustering over these 500 different clusterings. And this can then additionally increase the robustness of such a biclustering. So let me give a little bit of an example of how this looks like now in an application here. And so this is based here on data from the Cancer Genome Atlas, and specifically data from glioblastoma multiforme patients. So glioblastoma multiforme is a severe brain tumor, in fact. In fact, has very bad prognosis. So on average, patients die it's about a year after the initial diagnosis. And this disease has also a certain heterogeneity within it. There's also, based purely on transcriptome data, a known subclassification of this disease. So this is the so-called Verhaak classification. But of course, having this classification and having this data, of course, then also is an ideal test case at first place for looking in how far a multi-omics-based approach could potentially give additional insights here. And what I should mention here, one of the issues with the Verhaak classification is that indeed shows indeed also reproducible molecular subgroups, but these are not prognostically different. So which then in essence has a certain limitation then for clinical application. So what we now did here is to apply PathMe to this data. Multi-omics data here, so using DNA methylation, transcriptome data as well as CNVs jointly together. And on the right-hand side here, you can see a TSNE plot, so a two-dimensional visualization of this clustering which we obtained, so which indeed then resulted into 4 relatively well-distinguishable subgroups. And these subgroups you can then further investigate. So we can indeed then look whether there's a certain difference in prognosis of the disease, so specifically progression-free survival, which indeed in this case here shows a difference. And specifically there's one subgroup here standing out, this green one here, which indeed, and this is quite interesting here, shows an enrichment for IDH1 mutations. And in fact, it is known that IDH1 mutations are a positive prognostic factor in glioblastoma multiforme. So this perfectly makes sense. Furthermore, what we could establish is seeing differences among these clusters in another known prognostic factor, namely MGMT methylation status in gender, which is also known to be show differences in with respect to prognosis. And indeed, there was also differences in these clusters when we looked for enrichment with these Verhaak subtypes, so this earlier established and published subtype, all purely based on transcriptome data. So that means overall this subgrouping which we find has relationships to this Verhaak classification, but is not identical. So what is now interesting here, and as I said, so one in particular difference is here that here, in contrast to the Verhaak classification, we indeed see there also difference in progression-free survival of patients. So now one thing that is indeed interesting is that you now can dig more into detail about each of these clusters. So you can investigate based on this sparse non-negative matrix factorization. which pathways actually contributed most to each particular cluster, and more specifically also which data modality contributed within each of these data modalities most, these pathways most. And this table here just gives you an impression here for that. So for example, you can here see that subtype 1 here is, for example, associated to IL-23 mediated signaling events, and subtype 4 to signal events mediated by HDAC class 1, and so on. So let us here, for the sake of an example, pick one of these clusters, cluster 2, for further analysis. So cluster 2 here, so this is the red one here, showed a particular poor survival, in fact, of patients. And what we found here, and what is also shown here in this table here, is that this cluster, in fact, shows a association to PDFG and TGF-beta signaling. And both of these are, in fact, well known in their role in GBM pathogenesis and prognosis, as well as progression, and in fact also discussed as targets, in fact, for glioblastoma multiforme currently. So now one of the next questions then that one can ask is which molecules actually contribute most to these pathways. So let's take here at first place PDFG-beta signaling. So what you can now do is one can calculate so-called Shapley additive explanations of our pathway scores. So that means we calculate in how far a certain omics feature of interest, so let's say here this first gene here, in the case of transcriptomics data, transgene 1, how much this would on average change in all subtypes, subsets of features over transcriptome data, the pathway impact score. And so this score here, so which is here denoted by phi I here, so this is in essence here the so-called SHAP value. And this can be calculated even for each individual patient. And what is depicted here in this bar plot here is the median of these impacts, these SHAP scores, then over all patients that belong to a particular cluster, in this case here, cluster 2. So that means what we can now conclude from these plots here is that indeed PDFG-beta signaling, or the PDFG-beta signaling score, in essence, multi-omics pathway score, is impacted mostly on the transcriptome level by this gene transgene 1, which is a member of the calpunin family of actin bundling proteins involved in regulation of cell morphology, motility, and cell transformation. And in fact, it is also discussed as a candidate prognostic biomarker promoting tumorigenesis in human gliomas. So that means the fact that we find this gene here in contributing to this cluster here, which has a particular poor prognosis here, from that perspective makes sense. So this is another example here from a pathway, in this case the TGF-beta signaling pathway that contributes to cluster 2 here. And what one can do here is a similar type of analysis with respect to SHAP values. And what is shown here on the bottom here is, in this case here, showing also the transcriptome data or the influence of different transcripts of CPGs as well as microRNAs. And for example, if I just here pick the top CPG here, which is the most contributing one here to this pathway. So then one can find that this sits in the promoter region of the gene AXIN1, which is in fact reported to be downregulated in GBM. and has a role in the protein expression and localization in glioblastoma. So also this seems to make sense up to a certain degree then it is picked by the model. So to sum up this approach and the potential impact of PathMe, so what we have here is an approach for quantitative multi-omics data integration on pathway level, resulting into a pathway level score. And this has several advantages. So it results into an effective dimensionality reduction of the data and saving of parameters compared to more naive approaches. And our approach here also leads to, as I hope I have shown here, quite interpretable results. And yeah, so what we have done so far with PathMe is to do an interpreted biclustering of patients, but of course the same type of approach could also be extended and applied to, in fact, supervised learning tasks in, for example, directly predicting progression-free survival, or of course also to other disease areas. So that means overall the potential impact of this work is a stratification of patients into more homogeneous subgroups. And this, of course, to link back then to what I talked in the beginning of, could then increase also the success chances of, for example, clinical trials. So now one of the things, however, and this brings me now to the second part of my talk, is that in reality, this is a TCGA. So the data that we used here for PathMe is just one study. But it is, of course, essential always in medicine to test the generalization of any of these findings across multiple studies. But one of the really big challenges in medicine is the access to data to do that. And one should really see here the point that specifically in Europe, we have very strict legal constraints for using patient-level data. So that means there exist really silos of, I would say, of different patient-level data at different organizations. And it is very difficult to bring data from one of these silos to another one. So one of the ideas now that we had and developed already in the past in the autonomy project, in which we now also bring into a new project here, which has just started. So in the context of the national research data infrastructure in Germany, is the idea of simulating data in a realistic manner. So what does this mean? So that means so that we indeed train a AI model, and I will explain later on a little bit more how this is done, within a certain organization that has a certain real-world data available. And these models are trained in such a way that they allow for a mathematical representation of the real-world data. So that means there is a AI-based pattern learned somehow from these data. And these mathematical representations, these kind of rules in quotation mark, then allow us for generating synthetic or virtual patients. And this can be done up to any degree. So we can simulate 1 million, 10 million, whatever number of patients. Of course, one has to assess the quality of such synthetically generated patients. So this is here this backward error. And we do this by, in essence, looking in how far the simulated data fits the real world data. And I will show you later on a bit more how we do this. Furthermore, of course, one can ask, what is now, if I talked about data privacy and the issue of sharing data across organizations. So then, of course, one has to also ask, if I look into this synthetically generated data, is there any chance that from these synthetic data, I could somehow re-identify a real patient? If this was possible, so that would be a potential problem. Fortunately, there is a way how to mitigate this via a technique called differential privacy. So in essence, we can train this mathematical representation of the real world data in such a way that we can, with certain guarantees, mathematical guarantees, control the risk to re-identify any real patients from these synthetically generated patients. And I mean, I just want to point out here, so we have Memansa here also on the call. So this is indeed also part of the work here that Memansa has been done and contributed to. So let me just talk a little bit more about how we do this now. So in essence, when we think about clinical cohorts. So then we have to think about the fact that this data has on one hand, a number of different modalities that are captured, such as demographic factors, maybe a certain genotype or genetic variants, other type of biomarkers, maybe certain clinical assessments and so on. And then there's a certain time, of course, usually in many clinical studies at least. And of course, it is, I think, not unreasonable to believe that the values of a certain, let's say, biomarker at, let's say, visit 2 here are somehow influenced by the value that you have seen at study baseline, but maybe also by other factors here. And likewise, maybe that a certain biomarker, let's say, observed at visit 3 here might have also not only a dependency on the value of the biomarker at visit 2, but maybe also in the further past. So that means there are, and this is here meant by these edges, so there are these non-trivial conditional statistical dependencies between variables or variable groups on one hand, and then on the other hand, we have here these, indeed different types of variables which come together. And which we here can think about nodes or variables, or in statistical terms, we think about them as random variables. So now, one way to more formally think about this entire structure is Bayesian networks. So Bayesian networks describe, in essence, conditional probabilistic dependencies between variables. And on the right-hand side, I just here show a kind of toy example here, right? So let's assume you have you have a certain SNP here, and then maybe depending on the number of alleles here that you have here in the SNP, the value of the SNP, maybe another SNP here has different values here, and so there's certain conditional dependencies here. And maybe the ApoE4 protein then indeed depending on this value of this SNP here has other values, certain values. So you can think about these dependencies here modeled via conditional probabilities tables. This entire model is generative, so you can in essence simulate data from it, and you can use it to model also multimodal data. And you can also learn partially even based on data these dependencies here, while at the same time including knowledge. So for example, that SNPs cannot be influenced by clinical features. And you can also, and this is a further attractive possibility with Bayesian network, you can use these models to make predictions also over time. Let me just give you a little bit of a further information about what I mean by simulations that you can do. So one thing is indeed, you can do so-called counterfactual interventions or simulation of counterfactual interventions. So for example, you can ask what would be the case if this node 4 here in this network structure would take on a particular value? How would this influence the value of all other variables in this network? So for example, what would happen if I made people a bit younger or older? How would this affect all the other variables? And there's quite well-established theory, in fact, behind this, the so-called do calculus. There's a number of further issues, however, with Bayesian networks that I will not go into here into more detail in the interest of time. But at the end of the day, so what we did to account for all of this and how we brought this all together now is into a new type of model, a so-called variational autoencoder modular Bayesian network, in short WAMBN. And the idea behind this approach is really that we, on one hand here, discriminate different variables in the original study into modules, into groups, which are highly interpretable and which can also describe time dependencies in the data. And on the other hand here, within each of these modules, data can be encoded in a low-dimensional space via the technique of autoencoders, which I have previously described or briefly at least touched upon. And here we use a specific variant of these autoencoders, which allow us, in essence, then to learn or to map the original data, in fact, to a statistical distribution, a normal distribution more specifically. So this entire approach here, WAMBN, is therefore a merger of Bayesian networks and variational autoencoders. It's a white box model, can model arbitrary statistical distributions, and it has really a number of advantages compared to other machine learning approaches. So the entire model at the end of the day can be trained in practice by initially defining these modules in the data, then training these specific types of autoencoders, and then performing a Bayesian network structure and parameter learning. And based on that, then we can then simulate or draw patients by first drawing from these modular Bayesian networks and then decoding the samples through the autoencoder. Just to give you an example how this looks like in practice here. So this is a figure from one of our papers here, and it is about Parkinson's disease. It's a study in Parkinson's disease, in fact, and you can here see that indeed with this approach here, WAMBN, we are able to get a highly interpretable network representation on one hand of the entire study here, which comprises also the time course of certain variables here, how different types of variables, so for example, lab parameters are influenced by others, how and different types of clinical assessments here, for example, Epworth Sleeping Scale and UPDS are influencing each other, etc. So you can really, on one hand, use this for data mining purposes, indeed. So for understanding variable dependencies, and here these little numbers indeed and indicate the level of confidence that we have in each statistical confidence in each of these edges. You can, more recently, we have also applied this, and this is also work here by Memansa Sod in the context of the Radar ID IMI project to understand even dependencies between digital readouts and clinical assessment scores. So for example, here, this is data from a company in the US, which developed a virtual reality game to assess cognition in Alzheimer patients, and one can then try to disentangle how does the score which they derive relate to more traditional clinical scores in Alzheimer's disease. As I said, so we are in fact quite carefully checking how realistic simulations from such a model is. So on one hand, we can use this for data mining purposes, as I said, on the other hand, to simulate synthetic data. And what we can do, in fact, is we can look into the distributions, the marginal statistical distributions of different variables that is simulated by these data, and on the other hand, even for correlation structures that we can compare between the simulated and the real data. And in fact, we can also use the simulated data as such to learn network structures and compare them against the network structures that we have learned from the original data, which indeed here demonstrated that if you use more and more synthetic data, that you then more and more approach the structure that you learn from real data. So this is another way how you can check how realistic this synthetic data is. And as I said, so last but not least, we can use this now to simulate what-if scenarios. So for example, we can ask, what if patients in this particular study had at the beginning disease scores that would be the same as patients having in a different study? Or what if patients would have been a bit older or younger? Or what if even they had received at a study baseline a certain treatment of which the effect we have observed in a different study? So all of these questions can in fact be simulated with this approach which we have here developed. To sum up this work here and the potential impact, so WAMN is indeed an approach here for highly realistic simulation of multi-modal patient trajectories. So it's in this case here not only multi-modal data, but it's really also patient trajectory multi-modal data. It's based on a merger, in fact, of Bayesian network and variational autoencoders, and we see a quite broad impact of this work. In fact, so on one hand, in data science, to facilitate data sharing simply between different organizations. So for example, one can use, and this is also the thought that we have in this NFDAI for Health project now, the approach to train an AI model within a data holding organization to generate synthetic data that then could be shared with data scientists who then could use these synthetic data themselves for several purposes. A, to better understand the value of these data at first place for the research questions, and B, potentially even to train AI models with these synthetic data also that could be tested later on real data. So this idea is something that in fact already in another field of research, in imaging, is at the moment appearing. So, and the other thing is really the use for clinical trial design. So we are really thinking that with such an approach, one can really play with inclusion-exclusion criteria of trials. One can try to simulate control arms of trials and then even share these control arms between different organizations more easily. And last but not least, the idea is to use such approaches for clinical routine. This is something that we are currently discussing with the University Hospital Lausanne. So basically, one could use a patient sitting in front. So imagine you have a patient sitting in front of you with certain characteristics, and then you can ask, show me, let's say 100, 1,000, 1,000,000 patient trajectories and summarize and plot summary statistics over these synthetic patients who have the same or similar baseline characteristics than me. Yeah? So basically you can get a feeling how the disease might develop over time. At the last point, I also want to mention that we have now also a approach in hand which does similar type of things also with continuous time data even. And I will not go into, in the interest of time, into more detail here. But in fact here, so this approach which we are currently writing up a manuscript about is also then allowing us to extrapolate data to time points which we haven't seen so far. So overall, and that brings me to the end of my talk, I think precision medicine has the big vision, the ambitious vision to bring the right drug to the right patient at the right time, right? And realizing this vision really requires to jointly consider all relevant data modalities. Otherwise, this is not possible. And this is only possible, on the other hand, if you use advanced data science and AI models. And one of these examples here, which I showed here, was indeed Pathme as an integrative approach for multi-omics based patient stratification. On the other hand, any of these models that we build need to be sufficiently validated internally. So that's always the first place, right? So, for example, is in TCGA, but then also externally, so which would at first place mean maybe a retrospective validation with another pre-existing study, but in the long term, then of course always a prospective validation. And this always then brings up this challenge of how to access according patient level data. And here, indeed, the generation of synthetic data then comes into play. And here I showed you as an example to address this some aspects of that via a dedicated AI approach, Wamben, for simulating multimodal patient trajectories. So with that, I would like to thank you for your attention. And of course, I would like to thank here also the people from my team, in particular also those who contributed to the work that I just showed here, and also my former group members. And yeah, of course, I'm happy for any questions.
Okay, thank you, Professor Holger, for a very interesting computational aspects of multimodal data analysis. And I can understand, because I am also working on this various kind of omics data and how difficult it is to handle the omics data because of the heterogeneity of the data, the increase in the dimensions that we would like to handle. So it's really difficult, but the way you are approaching to model this data is really very interesting. So it was really interesting to listen all these methods and models that you have developed in your group. So we have definitely some queries from the participants. So I will go one by one. We have a very first query from Janki, and she is asking, is multi-omic stratification of disease being used in clinical treatment yet, especially for diseases other than cancer? What do you think the challenges that stop current stratification from being applied to medical practice?
Yeah, that's a very good question. At the moment, multi-omics data-based stratification is something that's in research, but that is not yet in clinical practice, even not for cancer. So I know this, for example, for my wife, who is a physician, and also, in fact, treating cancer patients actively. So how cancer patients are treated at the moment is very simplistic. So they're looking for a certain number of key somatic mutations, and then they have a certain number of antibodies which they know that work if a certain somatic mutation is there or not, and that's it. Yeah? So it's a relatively simplistic approach, but it is known also from research that this approach is in fact oversimplistic. So now, of course, one of the key bottlenecks to bring multi-omics into clinical routine is the fact that this is at first place technically more challenging, that it's also more costly potentially. Yeah? So what is, however, of course, good to see is that there's a number of techniques now coming up. So for example, I know there's a collaborating group also within Fraunhofer who's developing multi-omics techniques even on single cell level, who can make this indeed more and more cheap. So at the end of the day, if this can be made at a sufficiently low cost, so then, and at the same time, it is not only in research, but also in prospective clinical trials, being able to demonstrate the actual benefit for patients. So then, of course, one can try to bring in such approaches also into clinical practice. But this will take time. So I'm very honest on that. So we are not there yet at all.
Okay, yeah, thank you. So as you were discussing about the NMF, that is the sparse non-negative matrix factorization method. So she is asking, why do you think is consensus SNMF a better method of clustering in comparison to model-based clustering and other clustering methods?
It's not necessarily better or worse or anything like that. So the reason why we choose SNF-based clustering in this particular application is that we have a relatively high dimensional profile of each patient. So a number of different pathways in this case here. Dozens, or it could even be hundreds of different, let's say, mechanisms, the potential disease mechanisms. And we know that only parts of these features play a role for the subclass, for the stratification. So we wanted to have, on one hand, a clustering algorithm that is able to deal with this relatively high dimensionality, and at the same time is also able to result into a bi-clustering. With something like a Gaussian mixture model, so typical model-based clustering, so this would not be possible. I mean, I really like Gaussian mixture models, right? So it's not that they're bad or good or whatever, right? So, but there would, for this particular question which we asked here, not the ideal answer.
Yeah, okay. Because sometimes we need to decide the model based upon the requirement of the data that we are analyzing. So accordingly,
Exactly.
we can change the model. Okay, there are some more queries, like one we have from Santosh Patra. First, he is asking about the application of students' t-test.
Mm-hmm.
What are various applications?
So what's I don't understand the question. So what is about students' t-test? What does he want?
He's asking about just application. So I can tell that, like, you know, student t-test is a very common test, and normally we do it within the groups. So we want to identify the exact differences and how significant is the difference between the groups. I think, Santosh, you can get this.
I see. So I mean, the t-test, or whether you can apply a t-test or not, of course, depends on the statistical properties of the data, right? So I mean, if the data has approximately a normal distribution in each of the different groups, then you can apply it. So for example, in our case, differences in age or so, right? So that you can test with a t-test, right? So otherwise, if there's a clear deviation from the normality assumption, one can use alternative testing approaches such as the Wilcoxon test, Mann-Whitney U test. Also for testing differences of survival curves, there are specific tests. So for example, log-rank test. And if you want to account for confounders, of course, which we did, in fact, so the group differences in our stratification are not only, so are in fact corrected for confounding effects of age. In this case, one has to then use a linear model or generalized linear model.
Okay, there is one more query from Janki. She's asking, what are the major challenges, according to you, in the simulation of data in terms of time and generation of synthetic data sets?
So the main challenge, so I mean, of course, any of these approaches requires at first place a certain level of understanding of the data. So this takes time simply, right? So this is not something like a plug and play approach. So that means when we start modeling data with this approach, we first need to see what do the individual features really mean? Can we understand a certain grouping or ontology of these features? What do we actually want to achieve with this simulation? And so all of this typically requires also a certain collaborative effort with our medical partners. So we are not doing this usually without our medical partners and jointly discussing the data with them. At the end of the day, these models, and that I should also mention, also depend in their quality on the number of data, on the volume of the data that you have. So since this is an AI-based approach, right? So any sort of AI-based approach is depending on the number of data that you have. If you have larger data sets, so then of course the model is better generalizing. So then it is a better model. And on the other hand, for very small data sets, fitting such a model does not really make sense. So of course, then the next question that is often asked by people is, yeah, how much is enough? So unfortunately, so there's not a clear answer from theory to that. So there are certain, we know this from statistical learning theory, so there are certain mathematical bounds, but they are very too loose, in fact, to give any concrete answers. So at the end of the day, how good a AI model is something that you cannot estimate as simply as you can do this for a statistical test or so with power calculation, right? So that means what you can do is you can start with a certain model, a number of data points that you think might be reasonable, large enough, and then you can later on, for example, calculate what's called a learning curve in order to estimate how good would have the model been had you have more data or so. So this is what you have to see. So every of these things, of course, come also with their downsides. On the other hand, what we can do, in fact, with the generative aspect specifically of this data is to understand if we had more patients than we actually had in our original data, how much would this, for example, affect the quality of, also the chance of, for example, finding a significant effect of, let's say, a genetic variant or so. So this is indeed something that we have recently done in a master's thesis project. So we had, in this case here, SNP-based data of certain SNPs, selected number of SNPs, and then indeed, in the original data, we didn't find a significant result, but then we asked, so how likely would it have been to find an effect given that the data was, for example, 10 times larger or so, right? So these type of questions then you can indeed answer.
Okay, Professor Holger, I just want to ask you one thing. As you mentioned about various kind of data that you were dealing with, like gene expression data was there, mRNA and miRNA expression data was there. Then you have SNPs and copy number variation data also, and other omics data also. So in your plan or in your work, which data you found the most difficult one for handling? Out of
I think, well, all of these data have their challenges, right? So I think, which are quite specific, right? So I mean, for example, take SNPs, right? So I mean, obviously, genetics is very difficult, right? So because, for example, if you think about how rare or not rare certain genetic variants is, this is a problem, right? So if you think about correlation structures or linkage disequilibrium between SNPs, right? So this is a dedicated issue with SNP data, right? So on the other hand, then transcriptomics data, for example, has also its own challenges, right? So because, for example, there you have a high variability of the signal, right? So if I take gene expression data of you in the morning or in the evening, it might already be a difference. So I think I wouldn't say there's this one data set that is particularly challenging. All of these data sets have their own challenges. And at the end of the day, one has to get experience with all of them to adequately address these challenges.
Okay. Here, Professor Sayal is throwing one interesting point. He's saying that I hope AI-based approaches will help us to reduce the cases of misdiagnosis, which lead to more than 2 million deaths every year. Huge medical errors.
So can you repeat which diagnosis?
AI-based approaches will help us to reduce the cases of misdiagnosis.
Of misdiagnosis.
Yeah, medical and healthcare.
Mm-hmm. Yeah, well, I would say that, so I think in general, of course, you can, one of the hopes is to use machine learning methods for more accurate, I would say, diagnosis. I don't know whether it's possible to find misdiagnosis in a certain sense. Yeah? So because at the end of the day, whether you find something or not depends on whether a person takes a certain type of test. Yeah? So based on whatever. Yeah? So maybe even based on, so for example, for COVID, right? So at the moment you might have heard so that there is indeed now even the discussion that you could detect this from simply coughing, yeah? So from voice. So, but of course, this is only valuable if somebody does the test. Yeah. So if you don't do the test, so then you cannot diagnose anything. Yeah. So I think the interesting aspect from my point of view about these machine learning based approaches is really more that you can extract at the end of the day a signal from data which otherwise for a human being would not be easy to detect. So let's take this voice example again. Yeah. So I think for a human being it is very difficult to hear a difference between the coughing of a COVID-infected patient and a patient that has another type of infectious disease and coughs. Yeah? So, but maybe for a machine, there are certain components of the signal that make this discriminatable. Yeah. So, and this is, I think, more the strength. Yeah, finding these hidden features somehow in the data that for human being are different to extract and disentangle.
Okay, there is one more query, Professor Holger from Dr. Hemant. She is asking, do you think that wet lab analysis is required for the data validation?
Yeah, so I mean, of course, specifically, I think in the systems medicine field, right? So for if you think about the modeling of, let's say, biological systems and pathways and so on with AI-based approaches, which we have also done in the past, right? So then, and then maybe you simulate certain interventions into the system and so on. Yes, this of course always requires a wet lab validation. Or at the moment, for example, they're using AI methods also for drug targets ranking, I would say. Also, for example, in COVID. And there we are also working together, in fact, with a lab within Fraunhofer, in fact, other institute, which then does certain validation experiments for us. So this is, I think, indeed in some areas really essential. So likewise in the clinical space, right? So as I pointed out, we need these external studies, right? So to validate findings from a discovery study or cohort.
Okay, thank you very much. And I think with this, we are ending this question answer session. So it was really interesting to listen to you, Professor Holger, and I can see Mimansa is also available now.
Yeah, I would just like to thank Professor Holger for presenting in the webinar and for the wonderful talk. And I'm really happy to be the bridge between the two institutes, and I hope we continue this further on as well. So yeah, thank you.
Yeah, definitely, Mimansa. Thank you very much. We are in this position now because of you. You connected us, and I'm sure Professor Holger will also be connected to us in future also. We are definitely going to plan for some collaborations, student exchange programs. And our students are already doing wonderful in your university. From like you were supervisor for Dr. Gurnur also, I think. So Gurnur was also your student. Then Mimansa, many other students of our BTech Bioinformatics, they are really doing good in your lab, in your institute. And we would like to continue with the same.